So before I start my talk today, I want to highlight one of my authors. So my second author here, Miriam, is actually a sixth form student who's been working with me on a Nuffield project for the past month. So these Nuffield research placements give students from underrepresented backgrounds the opportunity to undertake STEM research placements at universities. So she's helped collect some of the data that I'm going to show in this presentation. So supervising a Nuffield research placement is not only really rewarding and fun for you, but it's also really valuable in showing these students that university is for them and they can do it. So in the spirit of the talks we've been having about diversity and inclusion and engagement, I just wanted to highlight this scheme as something to look out for next summer, if that's something you might be interested in. So today I'm going to be talking about how we can unravel Eocene ocean ecosystems using fish teeth. So the Eocene was a time period 34 to 56 million years ago. And for the purpose of this talk, we can discuss ecosystems as essentially all the life that's happening within an area. So essentially today, I'm going to be talking about what lived in the ocean 45 million years ago. So my choice of ocean, others are available, is the Southern Ocean that encircles Antarctica. And within the Southern Ocean, there's two distinct zones. So the Antarctic zone here, which is south of 60 degrees, and the subantarctic zone. So the subantarctic zone is between this black 60-degree south line and this red wiggly line. This red wiggly line is called the Antarctic convergence zone, and its location is defined by currents. So everything south of the red wiggly line is affected by Antarctica and so cold, and everything north is much warmer because it's affected by the rest of the ocean. So in my research, I'm trying to understand how life in each of these two zones was different about 45 million years ago. So today in the Southern Ocean, ecosystems in this zone are controlled by a complex balance of dust input, CO2 input, and light input, all of which results in photosynthesis. Mixing then occurs, resulting in carbon falling down to the bottom of the ocean and nutrients being brought up. But looking into a warmer, fresher, and more acidic future of the Southern Ocean and the oceans in general, we're expecting to see more dust and CO2 coming in, higher sea surface temperatures, less mixing, which results in less carbon going down to the seafloor and more nutrients coming back up. So hence, looking back in time 45 million years ago when it was much warmer than today can help us understand how these changes in the future might influence these unique ecosystems. So how do we look at what lived in the oceans in the past? One way to do this is to look at the fossil record. So in my project, I use muds from the seafloor, which contain microfossils. So a microfossil is like any fossil you might see in a museum, but they're tiny. So mine generally range between 0.01 and 1 millimeter in size. So we have to use a microscope to look at them. So most of the microfossils I look at are plankton, and there's two types. So there's these green ones up here that live in the surface of the ocean, and these ones down here that live on the seafloor. So those that live on the surface of the ocean photosynthesize, much like all the plants do on Earth. And as they do so, they take in CO2 from the atmosphere and lock it up in their shells. Those that live on the seafloor can't photosynthesize because light can't get all the way down here. So instead, in order for them to exist, they have to eat what falls down to them. So we call this stuff that falls down to them organic carbon, but the simplest way of thinking of it is it's made up of things that have died and poo. So counting the numbers of these shells that lived on the bottom of the sea, we can make guesses as to how much of this organic carbon, the dead things and poo, that fell down to the seafloor. So in the subantarctic zone, at this time, we've got less of these shells. So therefore, we assume we have less organic carbon coming to the seafloor. But in the Antarctic zone, we've got much more of these shells, so we assume there's more carbon. So if we put that on a graph over time, on the x-axis, we've got millions of years before the present, going from 52 to 40 million years ago. And the y-axis, you've effectively got number of shells or the amount of carbon coming down. And you can see in the blue, the Antarctic zone pretty consistently has so much more carbon coming down than the subantarctic zone, with a couple of exceptions. So there's an exception here, an exception here, and an exception here. So there's another type of microfossil I look at in the sediments, and that's fish teeth. So by counting the numbers of fish teeth, we can make guesses as to how many fish lived in the water column above. So unlike the seafloor plankton, there's pretty equal amounts of fish teeth in both the subantarctic zone and the Antarctic zone. So we can assume there's similar numbers of fish living in the water column. But that's where things start to get a bit confusing, because fish are one of the major ways in which this poo and dead things are transferred down to the seafloor, because they often eat the stuff that's living up here. So surely if we've got more seafloor plankton, we should have more fish, but we don't. So what is this difference that we've got between the Antarctic zone and the subantarctic zone? So one hypothesis we had as to why these differences exist might be because there's different types or species of fish living in the water column. So to try and understand fish diversity, the Nuffield student Miriam categorized lots of different shapes of fish teeth. So these ones here are actually between 0.05 and 1 millimeter in size. So to categorize them, she looked at 18 types of differences in the shape. So an example of some of those differences are the differences in the triangle. So this one is a much shorter, squatter triangle, whereas this one here is much more elongate. Another feature she looked at is the root of the tooth, so how it would sit in sort of the gum. So like this one here is much flatter, whereas this one's got much more protruding roots. So one way to plot up all the data she collected is as a PCA graph, which is here. So because she had these 18 variables on each tooth, it's really hard to plot that up as a normal sort of scatter graph, because we'd need 18 dimensions of space to see any patterns. So instead, a PCA graph handily flattens all those dimensions onto one 2D plot. So in this graph, we've got the Antarctic zone in dark blue and the subantarctic zone in yellow. And effectively how large the clusters are tells you about how diverse the teeth are. So the Antarctic zone has a much smaller cluster with only a few outliers. So we'd expect that that zone contains less diverse teeth compared to the larger cluster for the subantarctic zone. So we assume that the diversity of teeth matches the diversity of fish species. That's a little bit of a simplification, but we're working in water depths in the past that are about 2,500 to 3,000 meters depth. And the fish body record for this time period in those locations is sparse, to say the least. So actually matching those teeth up to fossil fish is difficult. And it's quite hard to actually reconcile whether this range in diversity of teeth actually matches the range in diversity of fish species. So without that assumption, what our data probably means is that we have something like this happening. So we have the same number of fish in both zones because of the similar number of fish teeth, but in the subantarctic zone, we've got a wider variety of the types or species of fish. So in conclusion, somehow these differences in diversity between the subantarctic zone and the Antarctic zone results in differences in the amount of carbon coming down to the seafloor, feeding these seafloor plankton. We don't actually know why this is happening yet, and that's one of the problems I'm going to be continuing to try to detangle over the next year of my PhD. So thank you for listening. Any questions?